And that's because these targets can be very transient, right? If you have a high use area like a bed and breakfast or something that's using a septic system heavily for one weekend and then they're not using it again for a little while, they could be really transient when you're actually going to detect that human fecal marker in your water sample. So community-based monitoring can be incorporated into enteric studies in many ways. You can tailor the study design, sample collection, and you can revolve it all around qPCR, which is what we think is a good way to do it. And one of the real advantages of this is that you can archive those water samples as small tubes of DNA that you've extracted from the water sample, and that allows you to go back and test with different source tracking markers in the future if you wanted to test for something new, which I think is a huge advantage because you can never go back in time, unfortunately, although we should work on the time machine. So confirming CBM using qPCR is reliable and viable. This is what I want to end on, just talking a little bit about how we've worked towards demonstrating that community-based monitoring actually works well in this context. Because this is a big criticism that we often get, right? We're essentially comparing ourselves and saying that everyone in this room can do qPCR as good as somebody who's trained by the US EPA to do it in a lab somewhere in Michigan, where I don't know where the labs are. So how do we do this? Well, I want to focus on model C first. Okay, this is the one that we really want to move towards, which is making sure that there's interest from both sides, right? I have my research questions that I think are really cool. I want to answer them. Ron has research questions he wants to answer, and you guys all have questions you want to answer. And if I'm the one who's telling you what you're doing because I want to answer my questions, that doesn't help you, and no one's going to want to do it after, one season, right? So I think the way that we get buy-in to this kind of a program is that there has to be an exchange of ideas, and there has to be a relationship that's built where you are getting questions answered and we're getting questions answered at the same time, and everyone's benefiting somehow from this. That's what we want to work towards. So this map here, you definitely aren't going to be able to read all of it. The legend is the more important part, where the way that we set up our CBM program is to have this part be first, where we have consultation with stakeholders, and we discuss what the program is, why we're doing the program. We make sure that everyone's trained and methodologically capable of doing everything. And this is the white section where we have these sections completed in collaboration between the researchers and the community partners. That's all the white sections. The blue sections are completed by the central labs, which could be my lab or it could be Freshwater Solutions lab. We kind of view them as a quality control lab. And so these sections are where we have things happening where we are quality controlling the samples. So we make sure that, for example, 10% of the samples run by a CBM partner might be run at a QC lab, so we can make sure that we're getting the same results. So we can do sort of a results validation. And we can do things like spiking different controls into the pipeline, so we can give a blind sample to a community partner, and we know what that sample should give them, so when they give us a result back, if it's really off from what that is, we know something's going wrong in the method somewhere. The point here, though, is that then we have this whole pipeline where the community partner is doing a whole bunch of things like collecting samples, doing data entry, doing the DNA extraction. There's a bunch of feedback coming in with the quality control lab, the central lab protocols, and everything feeds back into this process here where we all get back together and we discuss results, we discuss some of the challenges, we discuss some of the strengths, and we reevaluate our question and we go back up to the top and we start all over again, making sure that we do everything again slightly differently with feedback of everyone after this process has happened. And so it's very circular. There's action stages, there's planning stages, there's evaluation stages, and then we go back and we do it all again. And that's how we want to organize these CBM programs, and we found that this works really well because it allows us to make sure that things are working properly. Some of the things that we have to test for are things like comparing between different machines. A core lab machine is very, very sensitive. It costs $40,000. The machines that we use for our CBM program are slightly less sensitive, and they cost about $4,000. So for 10 times less money, you get basically a 1 log loss of sensitivity. So whereas our core lab qPCR machines can detect 2 copies of a single gene, so you think back to that little slide I showed you, that little green line, I can detect 2 of those green lines with my lab qPCR machine. The least you can detect with one of the Chai portable machines is 50. Okay? Now, that's not really a big problem because we can control for all of that stuff, right? If we want to be able to detect samples that have really low concentrations, we just collect more sample, right? So if you need to detect 25 copies of your gene, and we know that there's around 50 in there, which might be pushing the limits of the Chai, we just collect double the amount of sample, and then you have 20 copies, 25 copies in there, or 50 copies in there, depending on what your sample you're collecting. What's important, though, is that the Chai performs the same as the core lab machines in terms of how it amplifies a target. So you can see these lines here and the R-squared values. Those are just technical ways of assessing the slope of the line and the relationship between these dots. The closer to 1 you are, the better the correlation is between your samples at these different dilutions where we have these known dilutions of a target gene. The point here is that the CHI actually gives us an R-squared value of 0.998, which is basically 1. That's like a ridiculously good relationship. And that's actually better than the rotor gene, which gives us 0.985. Both of those are fine. The point, though, is that the CHI performs just as well as the core machine. It's just less sensitive. And that means that if you were detecting, if this is the number of gene copy numbers that you were trying to detect, the CHI kind of falls off when you get down into this really low number. For something like an enteric bacteria study, that's not a problem, because if you only have 5 or 50 copies of Enterococcus, you're never going to do a source tracking test on that and figure out what's in there, because that's way too low to do a source tracking test. Okay? We also know that our participants who do the CBM programs, they're really good at doing qPCR. I think you probably actually care more about it than the people who do it routinely in the labs. You actually pay more attention to what's going on. But if over 25 replicates, if we do a blinded study where we just give a bunch of samples to a community partner and we get them to run qPCR, they have very good R-squared values, very good efficiencies, and everything matches very closely with a trained lab person. And that's really important, because a criticism of CBM, of a community-based approach, is that the variability is inherent in the community partners. And this is where we're trying to say, well, maybe for SECI-DEF assessments, that's a variation, but I would argue that that's a variation that would be inherent between any two individuals. But qPCR takes out the variability because it just tells you a value. So as long as you do the method properly to set up a qPCR, you will get these results routinely and consistently. And that's really important. So this is some data from our Swimmer's Itch CBM project, where these are just all different samples. Okay, so there's no really any x-axis, and this is just the number of cercaria that we've estimated in that sample using our little formula. What I want to emphasize here is that this black line that you see going up, that black line is the value for each of those samples that we get in the lab at the University of Alberta. So with Sydney, she's good at qPCR. And then these little dots are the values that Ron gets in the Freshwater Solutions lab. And there's a couple things that I want you to note. First, I'll just give you the statistics. So 65% of our samples are within the 95% confidence interval. Over 90% are within the 90% confidence interval. That's pretty good. And what I want you to notice is that our bigger variation, the more variation is at the samples that have more copies, right? So we see bigger variation when we have samples with more material that we're amplifying, okay? That's important because we know that even in studies where we've compared between regulatory labs, so this is a study, Abentier and al. They published in 2013. This was a study where they took a whole bunch of EPA laboratories and they got them to run the same samples off of the same filters over and over and over again at all these labs for different source tracking marker targets. What they essentially showed is that this is the interlab variability right here, and it's presented in a really complicated thing, so it's not worth for you to really wrap your head around that necessarily. This is the log 10 copies per filter maximum difference between the labs, okay? So the point is that the Enterococcus test, there's a 0.6 log difference. That's almost, let's say, a 10-fold difference between the different labs. That's the maximum difference that they have. That's a pretty big difference. Where our Swimmer's Itch qPCR test falls is around a 0.28 log 10 copies per filter difference. That's our maximum difference. If you were to map what that looks like, because I think a graph shows this better, if you were to map what that looks like on a graph, this would be the actual copy value that you would get. This is the actual copy number. So this would be, say, 1 million, 2 million, sorry, this is a billion, 100 million, 10 million, a million. The point, though, is that if you map what this log variation means to that actual number of copies in your sample, this is what it looks like. This is the variation that you would see in that sample. Okay, so this lower one would be your lower variation. That would be the maximum lower variation you see in that sample. This is the maximum upper variation you see in that sample. It looks a lot like this, is my point. Okay, when we have higher copy numbers in our samples, we see higher variation, but they always track with the same relationship, right? We always see higher in both cases, okay? And that's also important because that means that we can rely on the CBM partners. While they may not get the exact same value as us, the trend in their data is the exact same as our trend. And that's what we see even when we compare core facility labs with the US EPA. Okay? If they were to show you this graph the way I'm showing it to you here, it would look this way. This is the way it would look. Okay? So that's good.
And who's to say you're right and we were wrong?
Right.
I'm just kidding.
That's true.
Freshwater Solutions say, well,
What I should do is make this graph again with the black line being you.
I'm just kidding.
So I want to wrap up now. So implementing qPCR using a community framework, I think it works really well. And it works really well most when we have some controls in place to make sure that everything's working properly. So we have to make sure that training is comprehensive, and we've done that. We have YouTube videos, we have Freshwater Solutions around to provide direct training, hands-on training, and we have lots of written protocols. And we also refine those protocols routinely based on feedback from our CBM partners, because I think that's really important. If you read something and you're like, I don't know what this means, that means that it's not written right because you didn't understand it. So we should change it. Quality control also must be a really important part of the relationship. And this means that we routinely will have samples that are passed on to QC labs, that like Freshwater Solutions Lab, or even to us, where we run the same tests that you ran, and then we make sure that it all works and it all makes sense. Consistency in the training and methodologies is critical. So we know that the sample type, meaning the amount of copies in the sample, right? If you have a sample that has a lot of fecal material in it, or a sample that has a very little amount of fecal material in it, and the DNA extraction protocol itself, those are where the two main sources of error are in DNA-based testing for qPCR. So we need to keep those things as consistent as we can, okay? So what's our vision for all this? How do we see this playing out? This is, I think, where I think we'll hopefully spur some conversation now. We see community participation at all stages to answer questions that are relevant to all groups. That's that's our main vision here. Is we want to partner with people to understand what the questions are at the different lakes in Michigan, and then we want to be able to take that information and design research that answers questions that we want to know, answers questions that you want to know, and make sure that we're doing that in a way that gets us enough data that we're not stuck with some monitoring information about potential fecal bacterial contamination, but not answering an actual question that helps us learn about how we might be able to maybe mitigate that fecal bacteria, or when is it most risky. We want to accomplish research while answering relevant questions related to the environment and to health. And so I think this is important. I think this is an opportunity for us to not only address environmental issues that might revolve around fecal point source or amount of fecal material in a lake, but we can also partner up with regional health authorities to actually contribute data to their monitoring program. 200 beaches are part of the Inland Beach Monitoring Program. You guys could easily monitor another 100 beaches as a CBM project and probably provide backup data or data for new beaches as part of all this that can be contributed into the actual health program. This is how we envision this working in Alberta. In Alberta, we've had lots of interesting things that I can talk about that, but I won't go into that right now. We want to democratize water monitoring to communities that are impacted directly by the state of their water. So we want you guys eventually to be able to design your own water monitoring programs. If you have a qPCR machine and you know how to use it, and you got all the things you need to know how to run it, then you can do your own monitoring. You don't need us. Once you know what you're doing, you can eventually become autonomous and you can run your own programs. You could be a QC lab and then more lakes build off of you. There's lots of possibilities. One of our big things in Alberta, and I think it's a big thing here, is that one of the ways we see this type of a program being sustainable is to integrate this into schools. We think that if you train a teacher how to do this, they'll train kids how to do it. It allows you to build water literacy into educational programs, which I think is really awesome. It also allows you to integrate really interesting genetics and biology into a curriculum at an interface and a stage where they're probably learning about cell biology and genetics. So you can teach them about how it works in a cell, and then you can show them how it works in a tube. And you can actually get them to do it in a tube, and they can run a sample that's relevant to your lake. And so this is a big thing for us. We've partnered with a group called Inside Education in Alberta to do this, and our plan is to try to have about 20 qPCR machines in schools in Alberta next year so that we can run CBM programs for water monitoring through schools. Our goal really is to supplement state-supported testing in some cases to expand the reach of existing monitoring programs. So that's, I think, something that's really worth having conversations with your regional health authorities about. And we think that there's also an ongoing education here piece that's related to the value of advancing water science. And I think we are all here because we're passionate about water. We're interested in understanding about how we can improve the health of our water. And I think that this is one way to do that. The more we know about these different types of things that impact our water, the better we can manage water. So this is a way that might look, where we have our community partners, and they're running data and collecting data. They get qPCR values with machines, and those qPCR machines are providing data that gets sent to the core labs or our lab at the U of A. We then do a bit of a data interpretation, make sure that everything looks good, but that data is passed right back to the community users. And if there's an interesting data point that might result in a beach monitoring issue, what we could do is pass that data right over to the lab scientists that are related to Michigan Public Health, for example. They can go and actually collect the samples they want to collect from places that are targeted by the CBM program. So you're basically targeting the regional health groups to collect samples at places that are the most relevant for a health collection. And then they do their data and report back to the people as well. So this is one of the models that we're following in Alberta for our enteric studies, is to engage with our health group, Alberta Health. And so we actually monitor now for most of the cyanobacteria and some enteric bacteria in Alberta, and do it exactly this way, where we provide data and we spill over any data that looks problematic from a site to the Alberta Health, and then they do their own tests. And so this lets them better manage their resources, because otherwise they'd be testing at lots of different places that don't have cyanobacterial problems. This way we can tell them where we think there's a cyanobacterial problem, and they go and test there only. So it's a way to really maximize resources of the health authorities. And that's where I'm going to end. Sorry I went a bit long. So these are all people that have been part of this, and there's many more that have been part of this. We have lots of funding agencies. In particular, I just want to mention Alberta Innovates, that through their funding, we've really built this program very comprehensively in Alberta. And that has really been allowing us to leverage a lot of that expertise and even some actual qPCR machines and infrastructure to come out here to Michigan, because we think this is where there's a potential for a whole different type of CBM program to be evolved. And so that's where I'll end, and I'm happy to take lots of questions. I think there'll be lots of good discussions. So thanks very much, everyone.